Good morning, everyone. Thank you for joining us so early on such a rainy morning. Welcome to the inaugural Diana Koh Breakthroughs in Cancer Learning series. Before we begin, please note that no food and drinks are allowed in this function hall. Thank you. And now I would like to invite Prof. Ashok Venkitaraman, Director of the Cancer Science Institute of Singapore and the NUS Centre for Cancer Research, up to say a few words, please.
Thank you, Hema. Good morning, everyone. As Hema said, let me simply again echo how wonderful it is to see so many of you out so early on a rainy Saturday morning. I really appreciate your support, so thank you for coming. I think all of you know why we're here, and I hope that this is the celebration and the beginning of something quite important in Singapore. It's been made possible through the Diana Koh Fund, and particularly through the generosity of Janet Lim, who is sitting here at the front, and whom we really thank and appreciate for having initiated this award. So Diana Koh, after a long and successful life in Singapore as an eminent member of Singapore society, willed her bequest to charitable purposes, and as Janet has told me, she has spent a lot of time trying to understand and consider what those purposes should be that would best reflect the aspirations of her cousin Diana Koh. We're fortunate that she had chosen cancer research, and our institute, Cancer Science Institute, National University Cancer Institute of Singapore, and the NUS Centre for Cancer Research at NUS as one of the beneficiaries as a group. So we have a lot to thank Janet and the estate of Diana Koh for. So please join me in thanking them all, really, on this very important occasion. This event has come through the work of a number of people. Obviously, the Cancer Science Institute admin team, ably led by Jerry Leong, have been instrumental in publicizing and organizing this event. So thank you guys very much for a job well done. I'll tell you that even in advance. A number of people in the Cancer Science Institute and throughout the faculty have been engaged in trying to find the right ways to use the Diana Koh bequest. And as you know, you will be hearing later that we've not only awarded prizes for the best research achievements from amongst our younger investigators, but we will also be awarding prizes for the best achievements, again, from amongst our younger investigators, students, postdoctoral researchers, in an effort to encourage and laud their efforts. So you'll be hearing more about this later. But let me turn for a few minutes to elaborate on what I set out at the beginning. This is a very important event, and I hope, and I'm sure you will all hope, that this marks the beginning of something which is new and important for Singapore. As you'll be hearing later from myself and from my colleague Anand Jeyasekharan, cancer is very common across developed societies. Singapore is no exception. But as the incidence of cancer rises and rises, the public sadly remain not very well informed about the causes of cancer, how to detect it early, how to get it treated, and what the socioeconomic impacts of suffering from cancer can be, not only on the patient, but for the people around him or her, and for society at large. So there's an enormous task that we have in Singapore for public education, for alerting people amongst the public as to what cancer is all about, so that they can understand the disease and make the appropriate decisions, whether it comes to treating it, preventing it, or living with it. And that important initiative has again been made possible through the generosity of the Diana Koh bequest, and is reflected in this learning series, which aims to present over coming months and years a series of lectures where people like myself, who have spent a long, long time thinking about and studying cancer professionally, can actually communicate some of our experience and some of our understanding to members of the lay public. So I do hope all of you will accept my thanks for being here, but also spread the word, because this is a really important thing that we all need to engage in. Cancer is not going away, sadly, for the foreseeable future. We need to do something about it, and this cannot simply be done in hospitals or research laboratories. It needs the involvement and the understanding of members of the public. So please spread the word, and let me end again by thanking Janet and the Diana Koh Foundation in particular, and all of you for being at this event.
So thank you, Ashok, for the welcome address. Also, I guess to just add on a bit to the thanks, like Ashok said, in terms of the admin support, but also on both sides. One is from our side, but also in terms of on Janet's side, from the, please forgive me if I'm, from the ACF as well as the CFS, the Community Foundation of Singapore and the Asia Community Foundation. And we have Joyce and Martin here, so thank you so much for working with us this last year. We look forward, we look forward to working together more. And again, like Ashok said, thank you to everyone for coming. It's wet, it's Saturday. But also I want to thank everybody who did apply for the grant as well as for the prize. And we hope that all of you in the process, that you could use this to clarify your thoughts on the impact of your work as well as future directions. And that we hope that you learned something and that you had fun in this process. There were a lot of great applications. Unfortunately, we could only pick one or two, but we'd just like to say that the calls will be open for the foreseeable future. So please continue to apply, refine your applications, and we look forward to seeing them again next year. Okay, so thank you everyone who did throw your hat into the ring and try. Okay, so I guess now I'll reintroduce Ashok. So Ashok is one of the two speakers who we have today. And what I really love about listening to Ashok is that he can really distill down the important, very complicated scientific messages and simplify it that everybody can understand. So a little bit of background about Ashok. So Ashok trained in medicine at the Christian Medical College in Vellore in India. And then he went on to do his PhD at University College of London. He did his postdoc and he joined the faculty at the University of Cambridge. 1998, he was appointed as the inaugural Ursula Zollner Chair Professor of Oncology at the University of Cambridge. And since 2006, he was also director of the MRC cancer unit there. And he held both of these positions until 2020, when we're very blessed to have him come to Singapore to share his knowledge and expertise with us. So here, he's director of the Cancer Science Institute of Singapore. He's also distinguished professor of medicine at the National University of Singapore. He has many other appointments, which I'll not go into. But his work, he's really known for his breakthrough discoveries in terms of understanding how the information in our genes can lead to cancer. And he will elaborate on some of that. And he's really one of the pioneering scientists whose work spans from bench to bedside. He spun out a number of different companies. One of them, called Foremost, is involved in trying to drug what people have thought of as undruggable targets. So we really look forward to listening and learning from him today. Thank you, Ashok.
Yvonne, thank you very much for that kind and generous introduction. To the rest of the audience, please don't believe a word she said. It's a real pleasure to be talking to you this morning about a subject that I'm really passionate about and has preoccupied me for the last 20 years. In fact, my family often say, my kids and my wife often say that I devote more time and attention to thinking about cancer, what's in our genes and what isn't, than to my family, sadly. But I'd really like to take on this rather interesting and important problem, what's in our genes and what isn't, in the next 10 to 15 minutes or so. And I hope there will be questions from all of you in case I fail to make anything clear. So let me start by outlining the size and impact of the problem. As I said, the incidence of cancer is rising worldwide. These are some of the figures from Singapore published in 2021 in the Singapore Cancer Registry's 50th anniversary report. You will see a few things that I'll draw your attention to. First, the number of cases of cancer has been steadily increasing from the time these records started until now. And this is adjusted for the growth in population and for the change in the age demographics of the Singaporean population, which, as we all know, is tending towards getting older, which is the age at which cancer is more common. So this is not a problem, as I said, that is going to go away. It's a major challenge for Singapore and for many other countries stretching well into the future. It's been estimated, where I used to work in the UK, that over time, between 1 in 3 to 1 in 2 people are going to suffer from cancer at some point in their lives. Think about that. 1 in 3 to 1 in 2. In Singapore, the corresponding number is predicted to be 1 in 3 to 1 in 4. But again, these are enormous numbers. If you're standing in a crowd of 100 people, 20 to 40 of them are going to develop cancer at some point in their lives. Can you think of a more important health problem than that? Frankly, I struggle. I really think it is one of the biggest challenges that humans face in countries across the world. The second important point I want to make is that, as all of you are aware, cancer doesn't come, unfortunately, in one flavor or one type. It affects virtually every organ in our bodies. Here in men, here in women, in Singapore. And you will see, as I'm sure you're all aware, that you can have cancers of different organs ranging from the lung and stomach to the prostate or the breast in women. Each of these cancers is almost a different disease. And you're going to hear more about this from my colleague Anand in a few minutes. But each of them presents a unique challenge for understanding how they arise, understanding how to detect them early, and understanding how to treat them, tailoring treatment for individual cancers in individual patients so that the outcome is the best that we can possibly achieve. So let me turn now to the topic that I want to share some thoughts on with you over the next 10 minutes. I think all of you will be familiar with the concept that diseases, cancer being no exception, are a combination of what we call nature and nurture. Or put in scientific terms, the nature being what we inherit in our genes from our parents and from our ancestors, and the nurture being what we experience in our environment. And this rather vivid illustration, or I should say collection of illustrations, makes the point that nurture can involve the food we eat the air we breathe, our habits ranging from exercise, sleep patterns, and so on. So really, everything that we experience as human beings can affect disease. But we also know that encoded in our genes is an inheritance that determines whether we are going to be more resistant or more susceptible to to diseases. And deciphering how nature and nurture collude to cause important diseases like cancer is one of the major challenges currently in scientific research. Because without this understanding, we do not have much hope of not only treating advanced disease, but my personal dream to be able to intervene early to prevent disease or treat it at its earliest stages when it is not so difficult to cure. So, a word about genes. This is essentially what makes us us. And for the lay people in the audience, with apologies to the many experts I see sitting around, let me explain a little bit of how genes work to make us what we are. So as you know, our bodies are made up of trillions of cells. These are literally only microscopically visible, but they're everywhere in our environment. I'm sure you would have heard that, for example, each of us sheds from our skin every day hundreds of grams of dead cells that just come off our skin. It's what we have to vacuum or sweep up every day from our houses. So our bodies are made out of cells, and these cells are constantly growing, dividing, working to keep us intact. You know that the cells are organized into different organs: the heart, the liver, the brain. And what I'm sure you also understand is that each of these organs contains different types of cells that work differently to make these organs different from one another. So what I'm going to talk about for a minute or two is what happens under the surface. Under the surface, essentially, is a so-called instruction book in the form of DNA, deoxyribonucleic acid, which is an amazing biological macromolecule that sits in each and every one of our cells and carries a code, an instruction book that does two things. One is that it is inherited from our parents and therefore carries on the genealogy, if you will. But the second is the instruction book can be opened at particular pages or particular chapters, and the instructions can be read out to actually enable cells to do the job of functioning. So you see, this instruction book encoded in our DNA is really vital, not only to explaining why some people are more susceptible and others more resistant to disease, but it also is the instruction book that keeps us alive, because it's being read every second, every minute, every day in each of our cells. So I hope you can see the beauty of this hierarchical organization, from the instructions in genes, which lead to the regulation that makes our cells work, to the cells that form our organs, and the organs that form our body. There's an amazing symmetry, an amazing amount of information that we need to understand in order to really understand how to prevent or treat disease. What's made much of what I have to share with you and what Anand is going to share with you possible has been, over the last 2 decades, a breakthrough in reading the instruction book encoded in our DNA. Now, DNA consists of just 4 letters. Imagine a book, hundreds and thousands of pages long, with just 4 letters, A, G, C, T. These letters can be used to make words. These words can be written in chapters. And each one of us has this book in each of our cells. If you can't read it, you cannot understand life. And the breakthrough has been an advance in technologies which actually started with Fred Sanger's work in Cambridge in the 1970s and 1980s. He used to be the director of my division at the MRC labs in Cambridge. But Fred really started this off by developing ways to read these 4 letters in these strings of DNA that underlie our cells. You can see here that not that long ago, in the early 2000s, it cost millions of dollars to be able to read the book. Thank you, Mona. That's very kind. I'll keep it here. Thank you. It cost millions of dollars to read the book, so you could not go around reading the book in every individual for every disease. But look at this. I think many of you, perhaps those of you who work in the electronics industry, will be familiar with Moore's law, which gives you a sense of how fast transistor chips have moved. The cost of sequencing, of reading the book of life, has actually fallen even faster than the rate of development of new electronics. So this has been a revolution, and you can see that our ability to read these letters has led, from 2005, the first cancer genome read, to now literally thousands of cancer genomes, which are enriching our understanding and enabling us to treat cancer in very selective ways. My own work has been on one such family of genes. If you inherit faulty copies of either of these genes, they're called the breast cancer genes, BRCA1, breast cancer gene 1 and breast cancer gene 2, then you have a very high risk of developing breast or ovarian cancer in women, or prostate cancer in men, pancreatic cancer, hepatobiliary cancer in both men and women. So this is a cancer risk or cancer preventing gene. What it normally does is to prevent cancer, but if you inherit one faulty copy from either of your parents, then you're at very high risk of developing cancer. One amongst the more famous sufferers is Angelina Jolie, who actually inherited from her parents one mutant copy of BRCA1. And I think many of you will be aware that she has, throughout her life, had to fight to prevent cancer occurring in her, including surgical procedures and other preventive measures. If we can really understand how genes like this work, then we hope that we can intervene early to prevent cancer without having expensive, complicated, debilitating surgeries and such procedures. So my lab, many years ago in Cambridge, started by finding how BRCA1 and BRCA2 work. In other words, what do they do normally to prevent cancer? And when they are faulty, what happens to cause cancer? And quite simply, we found that they work as a repair machinery for DNA. Think about this book of life, as I said, written in our DNA. Occasionally, it will be damaged. Errors can occur in it. So there has to be a machine that constantly scans and reads this book and corrects any errors in it. That's how BRCA1 and BRCA2 work. We were able to show that these genes are essential to repairing the book of life, and without them, the book of life accumulates lots and lots of errors which lead to cancer. I'm not going to take you through this. But this just gives you an illustration of how, since we discovered these functions in the early 2000s, how we have gone from understanding how BRCA2 works to really resolving, literally in atomic detail. I'm showing you a ribbon of atoms within BRCA2 that we managed to decipher to understand exactly how it works at the atomic level or even within cells. It just gives you a sense of the time, the effort, the number of different disciplines from biology to medicine to physics to engineering that are involved in solving big problems of this kind. So I've given you a sense of how faulty genes can give you cancer, BRCA1 and BRCA2 being an example. But we know that cancer is not purely genetic. We know that the air we breathe, our habits, can affect cancer risk. How do we know this is a question that you may be asking. Here are two examples that I hope illustrate how we know. This comes from work done in the UK by Dahl and Hill in the 1950s. They studied a cohort, funnily enough, of health workers who smoked. Smoking at that time was popular. In fact, there were advertisements encouraging people to smoke for better sleep and better health. And what Dahl and Hill found was that smokers had a 30 times greater risk of developing cancers of the lung. Irrespective of their age, their gender, their genetic inheritance, you smoke and you are at risk from many diseases, amongst them lung cancer. And the size of that risk is not small. 30-fold is a really big risk. And interestingly, as you can see, once this became clear and people were encouraged to stop smoking, correspondingly, lung cancer rates have been falling. So this is proof, if you needed more proof, that exposure to cigarette smoke causes cancer, and you can prevent cancer by stopping smoking. The other line of evidence comes from a really interesting set of studies that I'll share with you. I'm sure you're all familiar with the concept of twins, yes? Two babies born at the same time, sharing a lot of their genes. So you can use twins to ask, look, what is the risk in identical twins of developing cancer? If it's all in the genes, then both the twins should have exactly the same risk. But as you can see here, even twins have different risks of developing cancer based on their exposure, based on their environment, based on whether they smoke, based on whether they breathe dirty air, based on whether they have enough exercise and the food they eat. And again, these studies show very clearly cancer is not all in the genes. Yes? I hope I've persuaded you. So I'm going to really finish off by giving you a few examples now from our own work of how we've been tackling this really important and elusive problem. How do our genes interact with our environment to cause cancer? And the examples I'm going to give you are from the genes that we have been studying for the last 20 years, the breast cancer genes, BRCA1, and particularly BRCA2. So imagine the problem. Many people, some of them inherit mutations in BRCA2. They have a very high risk of developing cancer. What is it that causes cancer? So I'll share with you two insights from our recent work. The first is we have found chemical triggers that can increase the risk of cancer in people who have mutations in BRCA2. This chemical trigger comes from chemicals called aldehydes. And I'm sorry to say, when you were making your way here this morning, if you breathed in any exhaust fumes from cars, you were exposed to aldehydes. If you used shampoo on your hair, as I did this morning, you were exposed to aldehydes. if you ate a very carbohydrate-rich breakfast this morning, a sugar-rich breakfast, you were exposed to aldehydes. So these aldehydes are everywhere. They're made within our body, but they're also everywhere in our environment. So this realization that aldehydes can actually cause or increase the risk of cancer was very important in our understanding. How do they work? So what we found was cells contain some levels of BRCA2, which is necessary to prevent cancer. Those are the yellow dots. Now, women who inherit one faulty copy of BRCA2 have less of these yellow dots. Still enough for normal function, but less than normal people. What we showed was that when you have aldehydes around, that reduces the levels of BRCA2, so it's no longer functional, and the cells then become predisposed to developing further faults in many other genes, which leads to cancer. When we published this work, it caught the attention of the lay press. I think many of you will be familiar with the wide circulation tabloid, The Sun, which has a circulation of over 5 million in the UK. It's not a broadsheet, it's not a very serious newspaper, but I had a call the evening after we published the paper from a charming young lady who introduced herself as the science correspondent of The Sun, to which my reaction was, I didn't know that The Sun had a science correspondent. But they did, and she interviewed me, and this was the result. First and last time I've been labeled a Cambridge Uni boffin, which is an old-fashioned word for scientist. So this, I hope, illustrates how commonly present chemicals around us and made in our own bodies can work with our genes to cause cancer. And more recently, here in Singapore, through the work of Lee Ren Kong, who is sitting in the front there in the audience, we found a further link, just published last week actually, between diet and cancer. And what Lee Ren and his colleagues in our lab found is that if you eat diets, poor diets, full of junk food, full of what are called glycemic foods that generate a lot of sugar in your blood very quickly, that can temporarily, only temporarily, I reassure you, all of those who like sweet treats, please keep going, but not too much. That can temporarily, again, inactivate some of our cancer-preventing mechanisms, suggesting that that is a missing link between how food or bad diet can actually cause cancer. And again, you can see by the multiplicity of colleagues who have participated in this work, by the different methods that we've had to use, how long and how difficult and how arduous is our journey to uncovering these links. But we have a lot of work to do to uncover more of the links between nature and nurture, between genes and environment. And I do hope that we will have further insights in years to come. But I hope that for you in the audience, I've left you with an impression of how our genes and our environment can collaborate to cause cancer. Thanks. There is time. I used up too much time.
Yeah, it's fine. Thank you, Ashok. So, are there any questions from the floor? No questions? So I can start. So I guess now that in the last, many decades, that we know more about the information that's encoded in our genes that we can't change, what do you think that in the future this could be used for, both ends of the spectrum, in terms of how we can diagnose cancers as well as options for potential therapeutics?
That's a good question. In other words, what does the future hold? Honestly, if I could imagine the future, I would be a very successful and very rich man. I can't. the pace of change in medical research is such that even those of us who work in it have difficulty in understanding what new breakthrough is coming next week, let alone in 5 years. But that said, please allow me to speculate a little bit about what we might be able to do. So I was very influenced shortly before I left Cambridge by a lecture given by Lee Hood at the University of Seattle, University of Washington in Seattle. So Lee had been for many years pioneering ways to understand genes and proteins in our cells, and he had made a complete switch from thinking about small-scale gene cells to thinking about large-scale, which is habits. And he proudly showed off in that lecture how he was monitoring his own physiological parameters through a little wearable device. And he said, you know, I developed a cold last week, and I knew 5 days before I developed that cold, because of the change in my physical parameters, that I was going to get sick. And I knew what to do in order to shorten the course of the illness. He went on and elaborated. He had sequenced his entire genome. At that time, it was still a bit expensive. And he said, look, I've inherited from my parents susceptibility to hypercholesterol, so I have to watch out. I have inherited susceptibility to several other diseases, so I need to change my habits. And he had done all of those things. So, Yvonne, the first answer to your question is, I believe that an era when genetic information of everyone will be available is not far off. In fact, in the UK, people are now beginning to think about whether every child born in the UK should be sequenced at birth, in order to ask whether they can be told, as they grow up, as new breakthroughs in medical knowledge come through, how to prevent and how to treat diseases that they may be uniquely susceptible to. This is science fiction at the moment, but I hope that the examples I've given you and the examples you're going to hear about from Anand about how genes in specific cancer cells can be used to treat that cancer mean that science fiction may at some point become scientific reality. Always remember the words of the Astronomer Royal in the UK in 1953. "Spaceflight is bunkum," he said. And 1 week later, Russia launched the Sputnik satellites.
Thank you, Ashok. Last chance, questions? Yes, Desi.
There's a mic at the front.
Thank you, Professor, for the presentation. So let's say, well, I am a woman, not let's say. So we will be afraid of breast cancer. And the example that you showed was the BRCA1 mutation by Angelina Jolie. So let's say I do have the mutation. It feels like definitely sentenced to have the breast cancer, right? So I will be very concerned of like, okay, I have the mutation. How can I prevent the breast cancer instead of like treating or cutting off?
Absolutely, yeah.
Thank you.
So that's an excellent question. Thank you for asking it. Right now, as I said, as illustrated by Angelina Jolie's struggles, all we can offer is, when women have completed their families, to offer removal of the breast, removal of the ovary as a preventive measure. That is not good. So what we are trying to work out is two things. One, as I illustrated there, what is it that triggers cancer in people who carry the gene? So as you can see, the risk is high, but it is not universal. It's not 100%. So a key question is, what are the triggers? And if we can find those triggers, for example, diet and other sources of exposure, we may be able to advise women who carry those genes, do this, avoid this, in order to help them to prevent cancer. But there's another development underway from many different labs, including some of the work that we're doing, which addresses the second problem. So when you inherit a BRCA1 or BRCA2 mutation, every single cell in your body has that mutation. But not all of the cells become cancerous. So if we had, say, a simple blood test that could say, look, in this woman who carries a BRCA2 gene, everything is fine, or there is a cancer developing. It's still small, but it's starting to develop. Then we could do something about it very early without having to resort to these. So the development of early detection tests that can find when very early cancers are developing in people who carry mutations like BRCA1 and BRCA2 is a major goal of research in many labs, because if we can do that, then we may have much more hope of prevention, even using new drugs, because we can administer those drugs when the cancer is detected, not very late.
Thank you.
Thank you. Any other questions? Okay, so yes, Sophie.
So how close are we? Sorry, thank you. How close are we then to maybe treating at the genetic level as opposed to treatments at the organ level?
Let me grab the mic. Another great question. Thank you for that. So how close are we, in case you didn't hear, to treating at the genetic level? You know, in theory, to be able to affect diseases where you inherit a gene that affects all tissues, you need to affect or correct the defect at the stage long before the baby is born, when you have a fertilized egg, really, because that's the only time that we, multicellular organisms, start off in a single cell, which can be cured. That time is coming, but at the moment it's far off. As you know, there are many, many trials all over the world, including an infamous one in China, where somebody without permission used CRISPR-Cas9 engineering to change two baby girls that were born, changed all of their cells. So it's possible, even with the technology we have. But it raises so many ethical and other questions. Can we begin to engineer the human race for good characteristics, bad characteristics, not for treating disease? Too many ethical concerns and regulatory issues mean that although the technologies are already possible, it's going to be very difficult, I think, at least for the next 10 to 15 years, to develop our societal framework, our ethical framework, to make gene therapy of this kind, engineering embryos, possible. So what can we do instead? One idea is to take cells in affected tissues which are most at risk of disease. That's much more difficult, but people, not our lab, are beginning to work to see if, for example, they can correct the BRCA2 defect in some of the stem cells in organs like the breast. And that might help to prevent the occurrence of disease. So you raise a very charged question. We can do it. We can do it now, but should we begin to engineer human beings? The insurance companies might love it, but I'm not sure the rest of us will.
Thank you, Ashok. Thank you, Sophie. Any other questions? Yes.
Hi, thank you so much. I'm from the School of Public Health. Yes, and I was curious if there are any kind of prospects for a public policy you can think of, like following these breakthroughs that you shared that tackle the environmental variables, so to speak, right? Such as the one about junk food or aldehydes. And so, are there any like public policies you can think of, or do you think they're, yeah?
That's another very good question. Thank you. I'm going to have to give you my cautious scientist answer, which is the same answer I gave the science correspondent of The Sun, who asked me, should we now be telling all our readers to avoid drinking alcohol, to avoid using shampoo, to avoid eating foods that cause this type of thing? And I said, look, we can only identify clues that tell us what's going on. And the research is a bit too early for us to be advising people how to live. That said, there are some very sensible and effective public health measures that I think most people, including yourself, would sign up to. Don't eat too much junk food. There's an epidemic of diabetes type 2 across Asia. over 10 million people with diabetes type 2. And the work that we've done links diabetes type 2, which causes an excess of cancer risk, through this mechanism. Get enough exercise and sleep. Eat in moderation. These are sort of sensible things that I think anybody would advise, but unfortunately we're not in the position yet of recommending public health measures. I hope that will come in time.
Any more questions? Okay. Thank you.
I do apologize for taking too long. But
No, go ahead.
No, I would love to. I'm apologizing to Anand.
Oh,
No, it's fine. We're free and easy today.
Great.
like I mean, a lot of times we hear about the dire side of the increasing rates of cancer, but on the positive side, maybe could you share some excerpts on like the great progress or like life saved in the scale that has been
Definitely,
achieved?
yeah, thank you. So when I was training in medicine, most cancer patients we were treating died for two reasons. One is that they were diagnosed at a relatively advanced stage of the disease, and there was very little that we could offer them because usually you couldn't cut the cancer out. But the second reason is the armamentarium of different therapies that we had, whether surgical, whether medical, was so small. When I was training, we had maybe a dozen drugs that we were using in virtually all types of cancer. All they could do, a radiation therapy, all they could do was to kill cells that were dividing fast. But now, with progress from many different labs over the last 20 years, because we now understand what are the genetic faults in many types of cancer, we have precision drugs that can treat cancers without side effects and clear them very effectively. One breakthrough, which started about 2005, is in my own field. When Alan Ashworth's lab found that inhibitors of a particular enzyme, it's called PARP, but don't worry about it, it's an enzyme in cells. But Alan and his colleagues showed that if you treat cells with BRCA2 or BRCA1 mutations with this drug, it killed them. And within 10 years, this drug had been approved, and it is used now not just for ovarian cancer in BRCA2 mutation carriers, but also for breast and many other cancers amongst these mutation carriers. It works in believe it or not, in as many as 50 to 60% of these patients. Half of them don't seem to respond. We don't know why. But those that do respond, many of them have been alive now for 10, 15 years, which again tells you how understanding the cancer genome has really opened new opportunities. We have also realized that the immune system somehow goes to sleep. The immune system is there to protect us against infections, against diseases like cancer, but when cancer develops, our immune system goes to sleep. Work of Jim Allison, first at Berkeley and later at the Sloan Kettering, showed, again about 10 years ago, that you could reactivate, reawaken the immune system, and that was an effective therapy, which again works in about half of the cases that you treat. So breakthroughs like targeted drugs, the PARP inhibitors, or immunotherapy, as Jim Allison described it, are big new advances that are leading to a revolution in our ability to treat cancer. So definitely I should emphasize the positive along with all the concerns.
So we're also open for more questions. So we don't have a strict time schedule per se. If anybody has more questions, we're happy. Sure.
I think there has been also some media focus on cases and stories of patients who undergo radical remission, i.e. cancer patients who unexpectedly and against all odds kind of go into remission. And I'm curious, how do you conceptualize and understand this as someone who's been in this field for so long? And do you think it's worth studying this kind of cases as well? Yeah.
I'll answer your second question first. There is no doubt in my mind that exceptional responders who somehow really respond well to therapy must be studied to understand why they respond well. the expert in this area, who's been doing work in this area, is Anand. So I'm going to defer to him in his talk and afterwards to answer your question. But I think it's definitely a very exciting area. I don't know whether you'd like to make some comments now or after your talk.
Well, I think you're asking about spontaneous remission, right?
Yeah.
Where it just goes away by itself. Yeah, and the thought is that perhaps, like Ashok said, the immune system wakes up somehow, and what may wake it up may be an infection, maybe something else that has happened to them. But that's the general thought about this. But yeah, certainly something worth studying.
Great. Thank you all very much for your interest.
So thank you to Ashok for the very inspiring talk, and thank you to the audience also for the very engaging discussion. So probably one last point about the public health perspective is most of the mortality for cancer patients really comes stage 4 when the cancer has spread everywhere, metastasized. So at least what we know you can do is to very early go for all your routine health screenings, tell everyone you love and care about whatever you can screen for. If you could do a mammogram, you could do a colonoscopy. Yes, they're not perfect, but they're there, and this, if you catch the cancer early, this would really improve your chances of resecting it and having a better prognosis. So probably that's something that even if it's not in place from a government regulatory perspective, that you can certainly encourage all your family and your friends and loved ones to go for. It gives me great pleasure to introduce my good friend and colleague, Dr. Anand Jeyasekharan. And Anand also trained in medicine at the Christian Medical College in Vellore. And then after that, he was awarded a Gates Cambridge Scholarship, where he pursued his PhD in oncology in the University of Cambridge in the Venkitaraman lab. So they go back a long way. And then Anand, after his fellowship, decided to come to Singapore, which was a great thing for the community. He came before Ashok came. And then, so Anand is a consultant at the National University Hospital, NUH. He's also assistant professor at NUS, the National University of Singapore, and he is principal investigator at the Cancer Science Institute. So his research group really focuses on understanding both the fundamental as well as translational aspects of the environment, tumor environment, which is what Ashok has been alluding to, about how the environment that your tumor is in can then affect how it responds to chemotherapies or other therapeutic interventions. So Anand is really the perfect speaker because Miss Diana Koh unfortunately passed away from sarcoma, and Anand is head of the sarcoma group at NCIS, the National University Cancer Institute of Singapore, and so we're honored for him to share with us his insights into this disease. Please welcome Anand.
Thank you, Yvonne, for that very kind introduction. It's a great pleasure to be here, and thank you again, Janet, and the Diana Koh Foundation. So usually, when you go to a rock concert, the opening act follows, it doesn't follow the main act. But over here, you seem to have reversed the order. So, no. So, this is actually the first time that I'm speaking after Ashok in any forum. I joined his lab 20 years ago to learn. That was my journey into science. And actually, I heard him speak as a medical student, and I was like, wow, I need to go work on this. But and then 1 month into my PhD, I remember thinking with my hand in my head, what have I gotten myself into? But it got better from there. And I enjoyed learning science, and it's really been a privilege to take this journey under his mentorship all these years ago and be here now. So our paths kind of diverged a little bit after I left his lab, because I went back to train as a medical oncologist and tried to see how we could apply some of these things, as one of the questions was asked earlier. How can some of these fundamental breakthroughs be used to change the way we treat cancer patients? And that's kind of where my motivation for research has been over the last several years. So I, as Yvonne mentioned, I sort of chair the Sarcoma group at NCIS, and so what I wanted to share today was perhaps something that we've been doing in this space, so to see what we can do that is different from how we've been treating this disease in the past, and see whether any of this could make a difference to our patients. I think Ashok already alluded to this, and I just wanted to highlight that if you look at the top cancers in Singapore in men and women, generally the big three which we describe are lung cancer, breast cancer, and colon cancer. So these are the most common cancers that occur in both men and women. You will not see sarcoma in this list. And that's really what I wanted to highlight over here, is that most of these cancers occur in major organs of the body that we describe, because they are continuously having cells that divide in particular ways, and they are also exposed to a lot of environmental pathogens, insults, let's say. But sarcomas are a sort of conglomerate term used for a set of cancers that arise from what we call connective tissue, or basically the bits of the body that hold us together. And this could range from bone to muscle to blood vessels to the lining of some of the outsides of the tract, the digestive tract, which is also made of muscles actually, nerves, fat tissue, all the stuff that is not an organ per se goes into this broad umbrella. And tumors that arise from this are called sarcomas. So you can imagine that one of the biggest challenges is that they are so diverse, they are so different. Each of them is so different from each other, and that they respond obviously very well to respond differently to different kinds of treatment. The way that we have treated sarcomas is similar to what we do for many other cancers, and it's typically a multi-pronged approach. The classic three pillars of cancer therapy are surgery to try and remove it, radiation when you can try and give radiotherapy directly to a tumor, and chemotherapy. And as Ashok mentioned, two general breakthroughs that have come up in oncology have been in the space of targeted therapy and immunotherapy. Sadly, these don't have many options in sarcoma. And that's one of the things I'm going to talk about right now, is that we still rely largely on surgery, chemotherapy, and radiation therapy. And the way we do this is in what's called a multidisciplinary tumor board. So every patient that comes into a large academic hospital, their case will be discussed at a meeting where we have a whole variety of medical professionals sitting in there, from medical oncologists like myself, my surgical colleagues, the radiation oncologists, different people who look at scans, the slides, to try and understand what's the best way to sequence these tools that we have to address this particular patient's problem. And in the National University Hospital, the sarcoma group was set up by my friend and colleague Mark Puhendran, who was the chair of the sarcoma group before me. And he's a surgeon. He's a surgical oncologist. He trained specifically in cancer surgeries. He set up the first version of this about 10 years ago. And since then, we've sort of reached a point over here, and I think the same is true in the National Cancer Center, down the road from us, that we are probably doing things at a level or a standard that is probably seen at any major cancer center in the world. So, what you get in Singapore is really on par with what you would get in New York or in MD Anderson or anywhere else. And that's thanks to really the sterling efforts of people like him. But it really takes more than even the doctors. It takes a whole village to tackle a patient with cancer. So this is a story of one of our patients from many years ago who overcame osteosarcoma to then go on to go on with her life and her career. And it's very heartening to see many of these patients in our clinic when they come back for follow-up. And they may be missing a leg, but they still have a lot to offer the world, and they often compensate in so many other ways. And it really is through a whole bunch of people that work together to try and see how they can affect that change, taking a deadly disease in front of you, trying to decide how do we take them through a period of intense chemotherapy, how do we take them through a very major surgery, rehabilitate them from that, and then eventually take them out, and continue the follow-up. But at the moment, sarcoma outcomes overall are still bad. And this is a curve which we use commonly in medicine, which essentially tells you over time how many patients sort of are dying from the disease. And you can see that, as you get a larger tumor and what we call a high-grade tumor, most patients sadly will pass away from this disease. And this is true all around the world. A small tumor that is low grade, you can see that almost most patients will survive. But as you go into the larger and high grade tumors, the case gets worse. And that is because of several reasons. Why is treating sarcoma challenging? It is to do with the fact that these tumors are often aggressive. It is due to the fact that they are often resistant to many of the treatments I mentioned. And also that, as Ashok mentioned earlier, often they are very painless in their early stage. They are, also located very deep within the body. They are located close to major organs, makes it difficult to resect them. And the diagnosis, is also challenging. I didn't write it here, but the other big thing is that they are so different. Each tumor is coming from, an osteosarcoma, which comes from the bone, is different from a rhabdomyosarcoma, which comes from the muscle, is different from a neurofibrosarcoma, which comes from nerves. So they're all very, very different, but they're lumped under this one common umbrella of sarcomas. And so the way that we have developed our treatments over the last 40 years has been somewhat limited because we clubbed all these very, very different diseases together, and we tried to find one thing that fits all of them. But the single one intervention that, like Yvonne was saying, early diagnosis saves lives with this. And so the one thing that for us is, even more than educating the public is actually educating our fellow medical professionals. Because the single intervention that makes a difference to sarcoma outcomes is a good first surgery. And so that's why we actually encourage our colleagues in some of the peripheral hospitals. Sometimes if you see a lump that doesn't just seem like a benign lump, you want to send it to a sarcoma specialist center because the first surgery has to be appropriate. It has to have the right margins. They have to decide whether to do what's called a wide resection or a radical resection. These things make such a big difference. And in NCC, they have set up something called the SPEED initiative, which is really an effort of their group. They seem to have this thing about sprint and speed. But their Department of Sarcoma, Peritoneal, and Rare Tumors have this initiative where they are trying to see how to get these danger sign tumors into the main center as quickly as possible so that an appropriate surgery can be done. And what else are we doing with these tools that we currently have available? I talked about the 3 pillars: surgery, chemotherapy, and radiation. And in Singapore, we are trying to push the boundaries of each of these as well. So one thing that my colleague Mark and the other surgical oncologists are doing in NUH is using 3D printed parts of the body to try to increase the ability of them for them to do a radical surgery. So this is an example of the shoulder bone. It's not a map of Africa. So this is something that allows them to then do a more sometimes a bigger surgery than they would have been able to do in the past, while still preserving some function. Or this is an example, we just did one yesterday where some of these patients have very extensive tumors inside their abdomen, and it's an area where the chemotherapy doesn't penetrate very well. So during surgery, we have a procedure whereby we can directly use what's called intraperitoneal chemotherapy administration. And it's literally filling the abdominal cavity up with chemotherapy and then swirling it around for a while and then taking it out again. It's the only time that oncologists like myself go into surgery, the theaters, and it takes a while because we have to get used to this whole new environment there. And the other pillar is radiation therapy, and Singapore has acquired 2 proton therapy machines. What does proton therapy do? It's a type of radiation which uses a larger charged particle than traditional radiation. But what that does is allows less scatter of the radiation, which means that it can be more precisely targeted to sensitive areas. And because sarcomas occur sometimes next to the spinal cord or next to the brain or next to vital organs, the radiation can be delivered in a little bit more, in a more precise fashion. And this is particularly important for younger people and children because the long-term effects of radiation are significant. I had a chance to go in and see behind the scenes of this beautiful proton therapy unit. This is about 1/10 of the size of the sort of the generator of protons in the back. So it's like the size of this room. So it's amazing that these things, and so this is sitting there under the base of NCC right now. So I talked to you about what we are doing with our current treatments that are available, the current tools. But as I mentioned, the problem is that we've been using this sort of one-size-fits-all approach. And I like this illustration that was given to me by my friend Ed Chow at NUS, which is we take a lot of very different diseases, we try to give them a treatment, and then we say, okay, some of them respond, some of them don't respond, and many of them have side effects. But really what we want to be doing is moving towards an era of more precise delivery of some of these medicines. For instance, can we first divide the patients up by some of these genetic characteristics that we had talked about, and then therefore give them subtype specific treatment? I'll turn to this part later. So this is an example of how we've been doing that. So this is a patient of mine, a young man, 26 years old, who had a stage 4 type of sarcoma. It's called an inflammatory myofibroblastic tumor. It's a very rare type of a rare tumor. So sarcomas are rare. This is a rare type of sarcoma, but he had stage 4 disease. Probably not long to live when we first saw him. And there's no real standard of treatment because it's too rare to conduct large trials. So this is a perfect example where you can then try and see, okay, what can we do to push the boundaries? And we actually did a genetic sequencing on him. So we tried to understand from the tumor sample some of these mutations that Ashok talked about in the genes, and to see if anything could be targeted for his particular cancer. And we found an interesting change, which is it's listed out here. It's called ROS1. Commonly seen in lung cancers, which this particular gentleman had. And you know that in Singapore recently, the ministry introduced something called the cancer drug list, which in a way to kind of cut costs on because cancer is such an expensive disease to treat that we didn't want people using, drugs all over the place. But unfortunately, in rare diseases, sometimes we do need to use drugs in an indication not for which they were originally intended. But we found that the cancer drug list did allow us to use one particular drug for many cancers, and it was called ceritinib. And it was originally designed as a different kind. It targeted a different gene, but it interestingly had a sort of off-target effect on ROS1, which is a related gene to this original target called ALK. And so I was able to start this young boy on this drug. And you can see here this is a PET scan, which looks at through the body. This is near his back, these areas. These very bright spots are all his tumor. This is normal. This is the back of the throat. And within 2 months of treatment, they all disappeared. And this was very heartening to see. And he, essentially, after many years of suffering from all these things, he had tried many drugs in the past. And it was only after sequencing this that we were finally able to get him something that really worked. And he's been well since then, thankfully, and hopefully he will stay well for a while. And actually was featured in the news this year, because he went on to start a restaurant, and it's apparently a very nice restaurant in Marina Square, a Korean restaurant, which he's been expanding his business, and he was very happy with all this, and he was also happy with the press because more people came to his restaurant. So this is an example of how these genetic events can actually or genetic changes can be targeted in a subset of tumors. And then I said we would go to the other end of things, which is here you're saying, okay, let's try and group some diseases together to see whether we can target them through a particular drug. But then there's one more step that we can take, because truly, as Ashok was talking about, when you have a change in a cancer that means that it cannot repair its DNA, the cancers evolve very differently in each person. So they've started, even though you may have some common characteristics, actually every cancer is different. And really you need a treatment that is unique to that particular patient. And that's what we mean by truly personalized patient-specific medicine. And that's what we'd like to do in the future. We've started doing some of that, and this is a collaboration with my friend and colleague Edward Chow, who developed a technology. Really, the concept is quite simple. Can we take the tumor out, grow it in the lab for a while, and throw different drugs on it and see what happens? And that's literally what Ed set up, and I set up a clinical study to try and see how we could get patients with a different disease. This is lymphoma, another disease that I treat. To get their tissue samples to the lab to kind of run these tests when we throw different combinations of drugs at them, come up with a prediction, and then see whether we could treat patients based on that. And this was a national study that we led across the country. And this is one of our patients who actually benefited from this treatment, and she had a very aggressive type of lymphoma that went into remission, and she's been well since then. And my colleague Valerie at the National Cancer Center has since taken on this study, this protocol, to apply it to sarcoma. And she started, again, a national study, which we will be joining as well, to try and see how we can take patients with the samples of sarcoma from the surgery, grow it in the lab for a while, and see whether particular drugs can be used for that unique case. And this is one of her patients, a young lady with a very aggressive sarcoma, for whom they found an unusual type of treatment that actually worked very well. So I think this kind of illustrates the principle of how we can go from from the more one-size-fits-all approach to a much more personalized approach with cancer care. And it's all stuff that we are already trying to bring in Singapore. What else are we doing? We're trying to now see how we can maximize the use of all our surgical samples, because some of these sarcomas are large. They can be 20 centimeters in size, and the pathologists look at the edges to see whether the surgeon has removed it all, but then the rest of it is thrown away. So we're trying to see what can we do with all that material to understand these diseases better. And these are what we call translational studies that we have set up to see what can we do. Ashok was talking about, should we be sequencing everybody? Well, we're trying to start by sequencing every sarcoma patient, at least, to see whether we can get sequence-specific information. This is a national study, a collaboration with a company as well. We are looking at features within the tumor that may predict whether these new drugs, these immunotherapy drugs, may actually work. It only works for a small subset of sarcomas, but they have certain characteristic clusters of immune cells. So we're trying to see how to find those better. We're trying to take out the cells, and in parallel to Valerie's efforts of doing the drug screen, we're also doing different types of genetic screens to see if there are particular pathways or networks that are important to keep that cancer alive. And we're working with colleagues in the Cancer Science Institute, like Dennis, who runs a facility to study proteins at high resolution and in a multiplexed way using something called mass spectrometry, where we're trying to see are there new drug targets that we can identify. And finally, we're looking at the environment of these tumors to try and understand how they respond to therapy, like Yvonne had mentioned. I'm also very excited by a new collaboration that we've set up with the A-Star group, where and Ashok is a part of this as well, where we're looking at trying to come up with cancer vaccines. So when we take out the tumor, can we come up with a sequence-specific vaccine for these particular patients that we can then give them to allow the immune system to prevent those tumors from coming back once the surgery is done. And all this is possible in a space like sarcoma, in a way better than it is in breast cancer, because actually it's such a rare disease and we don't have clear treatment plans for many of these things. This is what the NCC group are doing. It's a busy slide, but again, and thanks to Valerie for sharing this with me, where they are also setting up in parallel ways to try and take some of these tumors, put them into mice to kind of create personalized avatars of some of these patients, looking at how to make what are called organoids to be testing some of these drugs on, and they're doing a whole lot of molecular analysis on these samples as well. So you can see that as a group, as a country, we're all very motivated to try and see, because I shared with you some success stories, but we also have very challenging situations in our daily clinical practice. And it's heartbreaking to see young patients, because sarcoma sadly affects younger patients, more often. And many of my patients are in their 20s or their 30s. And when things don't go well, it's not nice at all. And so everybody who works in sarcoma is motivated to try and go do more. And we need more smart minds to continue to work on these things. So if any of you are considering, what you might want to do in the future, these are good things to do. The next real frontier that we are also very excited about is in the space of cellular therapy. So here, this is something called CAR T therapy, where you take a patient's immune cells, you modify them so that they can sort of learn to target a cancer cell, and then you put them back in. And this works very well in lymphoma. In sarcoma, we don't think this is the right approach, but we are trying to do something called allogenic CAR Ts, where we actually take the tumor, the immune cells from not the patient themselves, but from a healthy donor, and then try and expand them and teach them to have something that is maybe a bit more general for some of these kinds of tumors. And we are currently running a clinical trial with a group that has spun out of A-Star, where we've just started this, and this is running in NUH right now. So with that, I'll stop and thank the village that we work with. So these are all my clinical colleagues at NUH, everything from my medical oncology to radiation to surgeons to it's a big group, and this is the SGH group, again a large group of people that we all collaborate with. So this is just to give you a flavor of where we are with sarcoma treatment right now, both with traditional approaches, which we think we are on par with most places, and with what we are doing to push the boundary of care. So thank you all for your attention. Yeah, happy to take questions.
Thank you so much, Dr. Anand, for focusing your talk on sarcoma. Obviously, is illness of great interest to me. But I wish I'd listened to both of you, before, when my cousin was first diagnosed with sarcoma. I remember that, it was a really difficult journey, but the most difficult was knowing where to go and to understand a bit more about the illness and what are the options for treatment. So my question is, at that time we were quite far away from anything to do with cancer research, but I really was at a loss to know where to access. I didn't realize that so much was going on. So how do people in the public get to know about all these various pathways? I was looking around, even clinical trials, because we were so desperate, and it was that I said, let's try anything. The one thing that my cousin didn't want was to have the traditional, treatment, which were very invasive. And I tend to agree with her. Sometimes the treatment is worse than the cure. But if we had known about all these researches that were going on, and, I think there would have been, some hope, at least. So I'd like to know just how can the public access, all these expertise and all these latest research that is going on?
Thank you very much. Yeah, that's a great question, and probably one that requires more gray hair than I have to answer. But I think the short answer is that actually Singapore is very blessed, I would say, because you have very strong academic medical systems within the government funded infrastructure. So between NCIS and NCC, I think these are the two big cancer centers. Tan Tock Seng has a smaller cancer center, but many of the more complicated patients are treated at NCC or NCIS. And I would say, the majority of actually the general public directly access this, because they go through the polyclinic system and then they get referred to NUH or to NCC, and they will start off with, of course, standard therapy. But when we see that standard therapy is not working, we talk to them very quickly about a lot of these things. Paradoxically, it is those of us who may be a bit more, affluent and can actually afford private healthcare, that you actually have all these options elsewhere as well. And private healthcare has its advantages in that you have, faster access to things. You have sometimes, quicker access to senior doctors. Many things that the government system will not provide. But an academic center has a corollary of advantages. So I think somewhere in there has to be this balance between the private and the government-funded systems for people everywhere to know that these are the things that are available. And one of the things that we are doing now as a national consortium is to come up with some of these. We are working with a company that's interested. It's actually spun out from an oncologist in NCC, looking at how do we match patients to clinical trials, and how can it be made public in such a way that any patient might be able to just go to an app and see what are the trials that are going on around Singapore. But that's still at the level of clinical trials. It doesn't tell us some of these other things. So I think that's where this public-private partnership would be helpful. We actually tested that out in the setting of lymphoma. So I have my colleagues in the private sector who, when they had very challenging cases, they would sometimes send them to us for that drug testing thing, and then we would send the patients back to the private sector. So I think some of that education probably just needs to happen in the medical fraternity itself, and then in the public space would be having these apps and things like that are available. But I also end to say that the other thing that we do need to perhaps highlight to the public is that some of these traditional therapies, chemotherapy and radiation, they used to be much more toxic than they are now. So just as we've had research in how we are coming up with new therapies, we also have had a lot of research in how to make these therapies more tolerable. So we have certain, in the past, cisplatin used to be called cisflatin because it, pretty much wipes the people out. But we now have drugs that actually completely block the nausea that comes with it. The hair loss is still something that happens with many of the drugs, but we even have a protocol now in NUH with a scalp cooling device. So, patients have the sort of thing sitting there while they're having the chemotherapy to prevent some of this. So there's a lot of what we call para-clinical research also going on. And I think it's useful for the general public to know that, even some of these traditional therapies may not be now as bad as they used to be in the past. Any other questions?
Questions?
Thank you, Prof. Anand, for the presentation. A disclaimer, I know barely minimum about lymphoma and immunology, so my next question might sound a bit stupid. So you talk about the allogeneic therapy, taking the immune cells from healthy patients and then treat the lymphoma patients. I would like to ask, will that actually involve immune response from the lymphoma patients?
Thanks. No, actually, that study is for a broad variety of cancers. The lymphoma, the cellular therapy really started off in leukemia and lymphoma because you have a very clear target antigen, which is the B cell antigen, CD20 or CD19. So there, you take the person's own T cells, we re-engineer them to have a targeting construct that goes and attacks the cancer cell, and then you put this person's own cells back into them. Allogenic studies are where you take healthy donors like all of us, we take our blood, but then it has to be a particular type of cell that doesn't, shall we say, go and attack all the cells of the body, right? So it has to be something that is engineered in such a way that it is both safe, but is also targeted to something that is found on a variety of cancers. So that is an approach because for many reasons that is a more suitable approach also for cost and the time to when we can make it. Because you can imagine that to take a person's own blood and expand it and give it back to them, sometimes the cancer progresses within that time very fast. So having what we call an off-the-shelf product is an easier approach. But it involves those two key components. How do we make it safe so that it doesn't target the rest of the body? And how do we make it effective so that it actually targets the cancer? And there are some strategies that are in place for that.
Thank you for the clarification.
Sure.
So may I ask the next question on what do you think on the use of stem cells in treating cancer?
In treating?
Yeah, because stem cells also have the capacity to home to cancer cells.
Yeah. So that's an interesting question. And actually, one of our colleagues in N2CR, Fawn, he has a very interesting study going on where he has looked at the ability of stem cells to go and sit in a cancer to see whether we can use them to deliver chemotherapy directly at the cancer. So they've re-engineered those cells to be there at the cancer site, and then when you can give what's called a prodrug, which is the precursor of the chemotherapy drug, and then those stem cells have an enzyme that converts it into chemotherapy within the tumor itself. It's a very neat and elegant approach. They've tried this out, and they're doing a study in dogs right now because it's funny. I have a dog myself, and dogs get two cancers mostly: lymphomas and sarcomas, which are the cancers that I treat. So they've started this study in dogs first, and they're already seeing some very nice results over there. So it'll be interesting to see how that can be applied to humans. The slight concern is because they are stem cells, the worry is that they will learn from the cancer cell and learn to grow also. So how do we make sure that they are safe? That is the concern. But for some diseases for which we really have no other good options, and I think one of the things that they are trying to start off with is also brain tumors, because really glioblastoma is a tumor which has really no good options.
So I guess to follow up on what Janet was asking, so we talk about the more one-size-fits-all drugs like chemotherapy versus in terms of personalized treatment, and the cost of this is presumably quite prohibitive per patient. So I guess where do you see the balance, especially in a public health setting, to use the two? Because to me, it doesn't seem feasible that everybody is offered a personalized.
Absolutely. I think that's a great question because, as you know, the reason that the MOH came up with the cancer drug list was because the healthcare costs of the country were being driven up just by cancer and cancer treatment, and because that was leading to a rise in insurance premiums, a lot of things that go with it. So this is the approach that, for instance, the UK uses, where different drugs are licensed for specific indications. But that comes with the challenge that you cannot then personalize treatment. So how do we balance these two? I think the answer to me lies in having academic partnerships with healthcare, which means that, it shouldn't be the mandate of the government to subsidize what could be experimental therapy, unless it is under the umbrella of research. So when a patient wants to go through an experimental therapy, it should be under a protocol which is maybe funded through other streams than by the public healthcare dollar. if it is through either research dollars or it is through industry dollars or it is through some other system which can then facilitate some of this work so that, the patient may benefit, we benefit as a society, we are learning from all this, because it may not work for everyone. And it's a bit wrong to say that, government should subsidize things that may or may not work. So that is where I think, this balance could come from.
Any more questions? Okay, so I guess last question, which is to go back to what Ashok said very early on, that if 1 in 2 of us or 1 in 3 of us are going to be diagnosed with cancer, what advice do you have for people so that we die of something else? We don't die of cancer.
I think the answer is we learn to live with cancer. Right, because I don't think it's a problem that's going away. And actually, when I talk to my patients with stage 4 disease, often you tell them it's incurable, but I tell them so is diabetes and so is hypertension. You're not so scared of that. Why are you so scared of this? Because they are all diseases that will not go away. You have to learn to live with it. And our aim, I don't think, yes, prevention is the best strategy. If we can, you can prevent it. But I'm a medical oncologist. I don't deal with prevention. I deal with the disease once it's there. So then our scenario is, how do we help people live with the disease longer, better, and in a way that they're not affected by it. You want them to open restaurants, you want them to do things with their lives. So I would say that the answer is, from my perspective, is we just have to find ways to treat it in such a way that people can live with it.
Thank you. Let's thank Anand for a really great talk. So I think now it really gives me great pleasure to introduce Ms. Janet Lim, and she's very kindly worked with us over the last year. So I was checking, it was March the 30th, 2023, and this was our first meeting at Burnt Cones, which is this ice cream shop downstairs of the Cancer Science Institute. And then we walked up and we did an illegal lab tour, I suppose. So thank you for reaching out, and thank you for coming over. And we are really humbled by the trust and confidence that you had in us, even though you didn't really know anything about us. And we hope that part of these initiatives that we have established would also go some way to what you were saying, which is how do we reach the public and how do we put this information out there that, it's not just dependent on the one doctor that you see, but you can access this information on your own. And so it's quite surreal that from that day when we were brainstorming on things we could do together to be actually here at this event and to have gone through the first round of everything that we have done. So we are really honored that we can contribute, hopefully in some small way, to honoring your cousin's legacy. And so I would like to invite you to say a few words. Can we welcome Janet, please?
Thank you, Yvonne. Well, first, it gives me real pleasure to be present here today to witness the inauguration of the projects that, as Yvonne said, we started working on a year ago. I have to thank Prof. Ashok and Dr. Anand, for kicking off the first Learning Cancer series. And if today's talk is an example of what is to come, then I really feel that this has been a very worthwhile program. I've learned so much, and Professor Ashok, you have made a very complex subject very easy for the layman and for the public to access. I think this is what we need. I think, as you said, so much of cancer treatment also has to have the cooperation of the patient. And honestly, the lack of knowledge was something that was really frightening for me when I had to help my cousin with dealing with cancer. So I look forward to many more of these lectures to come. Now, I'm also very happy that we have today the first awardees of the two other parts of this package of 3 projects, which is the Young Innovator Grant and the Young Innovator Prizes. Now, when I was interested in giving grants for cancer research, I thought that it's actually maybe it's not possible because, as I understand it, cancer research is a massive undertaking that would require big institutional funding, and I just didn't think that the kind of relatively small grants that I'm able to give, lends itself to this kind of funding. But thanks to discussion with Duke NUS and also with CSI, I was made to understand that, in fact, there is a gap, in supporting young researchers because they are at the beginning of their career and are not yet drawn into this big established cancer research. And also, as young researchers, they tend to be more innovative and, dare I say, take more risk. So that sort of resonated very well in me. And so I'm so glad that today is the fruition of that ideal that we had worked together on. So I won't take up too much time because all I need really is for me to thank Professor Ashok and Yvonne for navigating me through the complex world of cancer research. And I look forward very much to, I know that, these small steps are very important in cancer research, but as I understand from today's talk, there is so much progress has been made. But if cancer is such a very complex disease where there is such specificity in the cause of each type of cancer and where eventually we really need individualized treatment, then, despite all the progress that is made, there must be many, many more research that needs to be done. So it seems to me like it's going to be, a need that will exist for a long time, and I hope that the contribution we make from the Diana Koh estate would help to contribute in a small way. But of course, my hope is that this research will lead to breakthroughs on which other bigger research can be built upon, and eventually lead to easier outcome for other cancer patients. With that, I want to congratulate also the winners of this year's award. Two, I think, for the Young Innovator Grants. And I don't know if you have their names, but maybe it's already. Oh, okay, yeah. And then the two prizes. I think this package of projects will help to encourage, not only just advances in cancer research, but what I also liked about the project is that it provides the platform for collaborations between different teams and so on. Because I think, as we have spoken often, that working on a complex subject like cancer, there's no point working in silos, and that this collaboration is so very important. But thank you very much for helping me to understand this difficult subject much better. And I want to also recognize the tremendous administrative support that must have gone towards making an event like that possible. In fact, I was telling Yvonne, I said, just looking at the 3 projects, it seems so simple, but I can just imagine the complex administrative work that must be done, to make it possible. So thank you to the admin staff who have made it possible, and I want also to recognize the Asian Community Foundation who has, helped me to manage this grant. Thank you again.
Sorry, so Janet, if you could kindly stay and, yeah.
Thank you, Ms. Lim. And we'd like to invite you to give out the Diana Koh Young Innovator Grant. So this grant is aimed to support budding scientists like postdoc fellows and young clinicians in order to kickstart the investigation of key knowledge gaps in cancer biology and treatment, as well as to create a platform for the cross-fertilization of ideas. So in no order of merit, we would like to call upon the awardees. Dr. Iliyanambi Sundaramurthy, for his project titled Understanding the Mechanistic Basis So for his project titled Understanding the Mechanistic Basis for PARP Sensitivity in Ewing Sarcoma. Thank you. And we also have Dr. Hao Ching Shen. For his project titled Potentialized Mitochondrial dsRNA-Induced Innate Immunity for Cancer Treatment. Thank you. Now we would also like to congratulate the winners of the Diana Koh Young Innovator Prize. So this prize was to recognize outstanding students and postdoc fellows who have made significant breakthroughs in cancer research. Again, in no order of merit, we have our first winner, Mr. Yu Heng Chua. So he is a PhD student at the Yong Lulin School of Medicine for his work in glioblastoma, a deadly brain tumor with limited treatment options. Next, we have Dr. Lee Ren Kong, a Lee Kuan Yew Research Fellow at the National University of Singapore. So his work explores the impact of cellular metabolism on cancer susceptibility and progression. So thank you, Ms. Janet Lim. And now we'll like to hear more from the Diana Koh Young Innovator Prize winners and what drives them and their research. So we'd like to invite Mr. Chua and Dr. Kong up to give a... Oh, okay. Mr. Chua first, please.
So good morning, everyone. First and foremost, I'm truly honored for receiving the awards this year. So in the next 5 minutes or so, I would like to take this opportunity to share some of the basic research we have done in the past 3 years, and hopefully you can gain something useful from the talk. So our lab works on glioblastomas, which is the most fatal brain tumors in adults. In Singapore, there are around 100 cases of glioblastoma being diagnosed every year. Although it's not so prevalent in Singapore, it is a disease that is worth our attention, as almost 90% of the patients will die 5 years after the first diagnosis. So if you look inside the tumors, there's a small population of the cells known as the glioma stem cells, which represents the most malignant cells in the tumors with stem cells-like properties. They are highly tumorigenic, therapy resistant, and invasive, contributing to both the disease progressions and tumor recurrence. In our lab, we use the so-called patient-derived glioma stem cell lines as a model system to study glioblastoma biology, and we try to translate our mechanistic findings into new therapy, which could one day improve glioblastoma patients' outcome. So tumor growth is actually a result of cancer cells undergoing uncontrolled cell divisions, and I would like to thank Prof. Ashok for giving a very good introduction about genetic materials. So the blueprint of life, also known as the DNA or genetic materials, is actually stored in these X-shaped-like molecules known as the chromosomes. And during normal cell divisions, the one and most important thing that the cells need to do is to ensure that the chromosome is separated uniformly into the two daughter cells. And if doing so, they are happy. And if the cells mess up this process, it leads to uneven chromosome numbers in the two respective daughter cells, and this can result what is known as an unstable cancer genome. This can actually stop the cells from growing and ultimately block cancer growth. So to circumvent this, the cancer cells actually contain a so-called traffic light system that can detect these errors and stop cell divisions in the presence of errors to allow the cells to restore this abnormality. So in our published work, what we have identified is that we found a new protein complex consisting of 3 proteins shown here, which could regulate this traffic light system in the glioma stem cells to impair proliferation. So what does this mean for the patients? So clinically, if the gliomas have low RNFA expressions, the cancer cells actually receive a green light to proceed with cell divisions. As a result, the tumor grows happily, but the patient suffers. Conversely, if the gliomas have high RNA expressions, the red light is on, cell division is blocked. Consequently, the cancer cells grow poorly, but the patients live longer. For the translational part of our studies, we found that you can actually mimic the effects of these proteins by treating the cells with these two compounds. And finally, we provided a proof of concept that these drug combinations can actually potently reduce the proliferation of glioma stem cell in vitro. For future work, we would like to use the latest nanoparticle technology for the targeted delivery of the drug combinations to the tumors to stop the growth of the glioblastomas. So thanks for your attention.
Thank you, Mr. Chua. I would now like to invite Dr. Kong up, please.
Hi, a very good morning. First, I would like to thank the organizer for this wonderful event, and secondly, to the Diana Koh Foundation and Janet for the generosities. So we have spent a very good morning listening to Ashok on how we try to avoid cancers, and we also heard from Anand on how to try to live with cancers. So I would like to use the next 5 minutes or so and to tell you my experience over the past 10 years in fighting with cancers, and particularly, what can we learn from the genes we inherited? Because I'm in the line of cancer research, so my peers always ask me, Liren, how can we actually prevent cancer, or how does cancer actually form? We know that as we age, the chance of getting cancers will increase. We also know now that for women with multiple rounds of pregnancy, the chance of getting breast cancer will increase. So for clinicians, when they deal with the cancers, they care about what are the subtypes of cancers, lung cancer, breast cancer, colorectal cancers, and with this, we then design radiotherapies or chemotherapies. And if you're lucky, the cancer will respond, and almost certainly, some cases or most cancers will definitely relapse and come back stronger. And over the decades of battling with cancers, as summarized by Anand earlier, we now know that cancer is more complicated than we think they are. Every tumor cell is different. And with this, we design targeted therapies or immunotherapies to better deal with cancers. Hopefully, the patients will live longer. We now even know that every patient is different. So why do I say so? Each of us, we actually carries one copy of a DNA, a pair of DNA, each from our parents. And if one copy of this is mutated or defective, such as in the tumor-preventing genes like BRCA2, as we heard from Ashok earlier, it actually increases the risk of cancer. But fortunately, these individuals will be fine only until the remaining functional copy of BRCA2 is lost throughout evolution, and this will lead to cancer formation. And such information and discovery has led us to development of a specific drugs to kill these cancers. This information has formed part of the foundation of how we see cancers over the past 50 years. But right now, we know that there are exceptions to this. Let's use BRCA2 as examples. So as a fellow trainee under Prof. Ashok Venkitaraman in this empire in Cambridge and now in Singapore, we work on BRCA2. 1 in 800 individuals that's walking around carries a mutated BRCA2 gene. And as we have known earlier, these mutations will lead to increased risk of cancers over time, particularly before the age of 45. And what I have discovered under the mentorship of Ashok over the past few years is that there's in fact a chemical strongly associated with bad diets and diabetes that could inactivate these BRCA2 proteins over a short period of time. It is still okay, but when these events happens frequently and over multiple times, it actually increases the chance of us getting cancers. So how do we make this discovery? In the laboratories, we use very sophisticated methods to look at the tissues, the malignant, tumor and non-tumor tissues in these BRCA2 carriers. And we actually found 2 things. First, we saw that the waste products that the bodies generated over the breaking down of sugars are actually accumulated in these breast tissues. And in the same tissues, we found evidence that the DNA has been changed. And adding 1 + 1 together, we show that how our cells uses energy and food actually influences and impacted how the tumor grows and evolved. And these findings actually has a broader implication to society. It is because it actually links to junk food that is high in sugars, and this junk food may turn off our body's inbuilt defense mechanisms, leading to cancer formations. And such finding has made to the international stage, as Ashok has mentioned earlier. And the most important thing is to raise awareness of how we deal with food, the environment, as well as our body, the gene. So, what are the messages I want to take home? Particularly for my boy over there who likes to eat sweet junk food, right? The adverse effect of consuming food high in fats and sugars actually accumulates over time. It's not just about stopping it over a few months. It's about a lifestyle, a disease that we have to live with. The poor diet, particularly those high in sugars or uncontrolled diabetes, could increase cancer risk. And most importantly, our genetic inheritance, understanding who we are and what is being carried in our genes, together with our lifestyle choices, carry valuable information that can influence the way we see and treat cancer. And with this, I would like to thank you for your attention.
Thank you, Dr. Kong. Could I invite Prof. Ashok up to the stage to present a token of appreciation to Ms. Janet Lim?
Thank you so much, Janet. I really hope that this will be the beginning of many, many more things. But thank you so much.
Thank you. And now we have come to the end of our learning series. Thank you everyone for joining us. And we have some refreshments outside, so please help yourselves. Thank you.